I'd like to thank the organizers for inviting me tonight to speak a little bit about how life might have begun on Earth. So, to contextualize what I'll talk about, I should say before I begin that my group at UCL, our ultimate goal is to build a cell ab initio, without any components from biological sources, and build this in such a way that it could have been built plausibly on a primordial Earth, and in such a way that it could potentially evolve to become life as we know it on our planet today. So this is a huge multi-faceted challenge, and I'm going to focus this talk on just one aspect of that. And I think that's one of the most important aspects of that, and that's the origins of genetics and the origins of information transfer in biology. So I'll give a little bit, a very brief, all too brief introduction to biological information transfer and the importance of RNAs in this process. It's probably noteworthy at this point. I didn't know exactly what Nick was going to talk about, but he mentioned ATP was the common currency, energy currency of biology. This is also a monomeric nucleotide. It's an RNA itself. And I won't talk about that in this specific talk. And then I'm going to move on to discuss how we can assemble two of the four canonical units, two of the four essential monomers to build information-rich RNA in a prebiotically plausible scenario, so without the requirement for any input from biological materials or enzymes. And then how these monomers might have come together to form the specific polymeric backbone of RNA and the specific polymeric backbone that we find today in biology. Okay, so before I begin, I'd like to put this talk and the ideas of this talk into historical context. So this is a timeline of life on Earth in billions of years. And here we are, we've just appeared on the scene right at the end of the timeline, moving back. We have dinosaurs, our first hard-shelled animals you'd recognize in fossils if you pick them up in your garden. We can go all the way back to 3.5 billion years ago in the rock record and find reasonable evidence for life on Earth. And we can find, it's argued about, but we can find fairly reasonable evidence in isotopic ratios of carbon, for example, for life as far back as 3.8 billion years ago on Earth. We know the Earth formed about 4.5 billion years ago, and we know it took a little while for the Earth's surface to cool enough to allow organic molecules to form. What we want to address is what happened in this gap here. After the Earth cooled, before life appeared on Earth. How did that transition occur? Okay, so we have a really beautiful theory that describes why we have diversity in life, and this is Darwinian evolution. So natural selection, mutation, change, and then survival of the fittest. But this really seminal theory says nothing about the origins of life. It requires a species to evolve. So it doesn't say anything about the event right down here, and you can't read this, but it says origin of life in that little green box. And this was something Darwin himself noted, and this was alluded to in the introduction to these talks. And Darwin recognized as early as 1863 in a letter to Hooker, but if, and oh, what a big if, we could conceive in some small warm pond with all sorts of ammonia, phosphoric salts, light, heat, he said electricity, not certain that's essential, present that a protein compound was chemically formed and ready to undergo Darwinian evolution, further change. So it's noteworthy here to say this is the 17th century, and protein to Darwin didn't mean what protein means today, Nick's big molecules of amino acids. This just meant a compound that was essential to life. So Darwin's saying here, oh, if we could think of a way that chemically a compound could assemble that was absolutely essential to life. And that's what I'd like to address here, maybe not in exact ways Darwin thought it would occur, but that overall concept. Can we understand how chemicals can self-assemble into a compound that's truly essential to life? And the compound we're going to think about, which is arguably one of the most important compounds in terms of evolution and information transfer in biology, is RNA. And Nick alluded to this in his slide of things that were conserved in biology. And the top 4 or 5, most of his slide involved RNA, the transcription, the ribosome, all of that's really important here. And what I have here is a description of the flow of genetic information, the flow of information in biology. And it flows from DNA with its now iconic Watson-Crick base paired anti-parallel double helix that contains our genes, our biological information. But if biology wants to use this information, it transcribes that. So it writes it out in the same language, but in an easier, more accessible form. And that form is RNA. And then it uses a whole series of other RNA molecules to translate that into a different language, into proteins. And I've got a video here, because this demonstrates this far more than I could possibly get over to you in 2 minutes, how important RNA is, how beautiful the processes are in biology. So this begins with post-transcription. That's a piece of RNA.
Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
Okay, so a really beautiful process. This is at the core of how biology is transferring information from genes into functional materials. The first thing it's doing is taking the genetic information in DNA and making that into a copy in RNA, something it can read, something it can use. This RNA copy is then read by a second set of RNA molecules. These are the transfer RNAs. They were the green molecules in the, or the green compounds in the video we just watched, and they have both RNA information, so nucleotide information here, and protein information, amino acid information, and they allow the translation of one form of information to a second in biology. And then finally, we have this hugely important machine, this molecular factory for building proteins, and this is the ribosome. This is the biggest piece of the ribosome. It's a crystal structure from the Stein's lab, who won a share of the 2009 Nobel Prize in Chemistry for elucidating this structure. And what you can see here is decorating the outside of this, we have in blue proteins. We have the business end of the molecule, the peptidyl transfer site, shown in red. This is where the amino acids are joined to form our peptide backbone. And all this in brown is all RNA. Everything around the peptidyl transfer site is RNA. This is an RNA machine. This is an RNA factory for building any protein that you need in your cell. These are so important that they are generally what's used, and they're so conserved to define the evolutionary distance between species on the kind of phylogenetic trees that both Nick and I have shown. They're really highly conserved, they're really essential, and they're built of these really complex molecules, ribonucleotides. So the question I have for this talk is, could these ribonucleotides that are essential to biological information transfer, could they self-assemble? Could a molecule as complex as this, with 4 information-bearing subunits built largely of nitrogen that have to be specifically attached by the correct nitrogen to a quite complex sugar that has to specifically have this 5-membered ring structure in the middle and 1, 2, 3, 4 stereogenic centers. So stereogenic centers are the things organic chemists quake about. These are really difficult centers to make. And usually, as an organic chemist, if you wanted to make such a center, you would borrow from biology. Biology is full of these stereogenic centers, and that's usually where we get our stereochemistry from in biology. And then finally, we have this backbone. We have a sugar phosphate backbone, and we have 2 options. We have the biological option, attaching the phosphate to the 5' hydroxyl and the 3' hydroxyl, or in theory, we could attach to the 5' and the 2', but we don't see this one in biology, we just see this one. So can we address where this kind of molecule with that level of complexity comes from in a scenario like this? And this is stolen from the Facebook page of tonight. But basically, the early Earth, with only the components available to us through geology, only the components available to us prior to biology, so no chiral pool. This means what I was talking about, stealing molecules from biology to build chiral molecules, and no enzymes, just using the intrinsic reactivity of really simple organic molecules. So this pursuit got off to a really good start in the 1960s. It was found one of the simplest organic molecules, just 1 carbon, 1 nitrogen, hydrogen cyanide, could oligomerize to give a tetramer, and that tetramer then could be quantitatively so all of this material converted into this material by UV light into this aminoimidazole, which could then potentially hydrolyze on to a second aminoimidazole. It was also found that these are dotted lines. This was low yield, but you could convert these aminoimidazoles into adenine and guanine. 2 of the components, 2 of those nuclear-based components, which looks brilliant. The problem with this chemistry is not only the really low yields here, is we get a plethora of purine compounds and a whole host of other compounds that are completely irrelevant to RNA. And there's no mechanism to select for these 2 that are important in RNA. So what about the sugar portion? Well, this is going even further back. So this is contemporary with Darwin. In 1861, Butleroff reported that he could take formaldehyde, again, an incredibly simple organic molecule, 1 carbon, 1 oxygen. And this is the solution we use to preserve dead bodies and organs in jars. This is a formaldehyde solution with a human brain inside. And if he mixed this formaldehyde with slake lime, so this is calcium carbonate that's been heated and then hydrated. So slake lime and this gives a really complex mixture, and I don't advise this at home, but it was standard practice in the 17th century. Butleroff tasted his reaction, and he found it tasted sweet. So he concluded he'd made sugars, and he was right. Analytical techniques and health and safety have moved a considerable distance since over the last 150 years. And now we can analyze this complex mixture of sugars, and what we do find is he's right. And ribose is produced in this mixture, but it is a very, very small proportion. So these two red arrows depict the proportion or the peaks that are ribose, and it's the area under these peaks that defines how much is in this solution. So a tiny fraction of what's there. So the lack of selectivity in both of these processes was seen to be bad, but not the final nail in the coffin for RNA. What for a long time was seen as that very thing, that the end of RNA in the context of the early life, was that if we had pure ribose in solution, one, it exists predominantly in the wrong sugar form, so the pyranosal form, rather than the five-membered furanosal form that we find in biology. But more damningly, only adenine can be attached to the sugar to give a very, very low yield of adenosine. And this is produced in a whole mixture, again, of all kinds of compounds that aren't found in RNA. So there's no selection for the molecules that are found in biology based on these chemical reactions. And finally, uridine, cytidine, and guanosine just can't be made through this process. And this was seen by eminent scientists in the field. And here's Leslie Orgel, who was mentioned by Nick slightly earlier, and Stanley Miller of Miller-Urey fame, that this just meant it was too complex. RNA couldn't be made this way. It was a step too far for abiotic, non-biological chemistry. And they went as far to say there was no basis that this could be achieved in organic chemistry. Now, obviously, I've set this up as a big challenge, so this is what I'm going to address, and I can provide a mechanism that we can solve this problem with. And what I can show you now is a synthesis of both of the pyrimidines, so cytidine and uridine nucleotides, the two of the ones that couldn't be achieved at all through this glycosylation procedure, and with a slight twist, so a slightly unexpected aspect to this synthetic procedure. We can take cyanamide, again, a really simple organic molecule, 2 nitrogens, 1 carbon. This can be condensed with a simple aldehyde, glycolaldehyde, to quantitatively give 2-amino-oxazole, this simple aromatic structure here. So all of the material coming in becomes this molecule. It's important we have phosphate. This controls this reaction, but that's okay because we need phosphate later in this process. Our ribonucleotides will in part be built with phosphate, so it's okay to have it there at the start. In fact, it's actually preferential. If we 2-amino-oxazole then condenses with glyceraldehyde in a rather spectacular reaction. Again, a near quantitative reaction to generate pentose aminoxazolines. Of these pentoaminoxazolines, 70% is the ribo and arabino forms. Each of those is only one stereochemical inversion from the beta-ribo form that's found in biology. So of those 4 contiguous stereocenters, in each of those molecules, we've only got one wrong. Even better, if you cool this reaction to 4 degrees, the ribo form crystallizes from this mixture, pure ribo in its crystalline form. And this graph tells us something even more exciting about this crystallization process. So nucleotides are chiral. We've come across this term a number of times. This means, like your hands, they have 2 potential non-superimposable mirror images. Unlike your hands, only one form is found in biology. So only the right-handed, I'm drawing it on the left, they're usually called the right-handed, but only the right-handed nucleotides. Our aminoxazolines, our riboaminoxazoline, also a chiral compound, and again has 2 chiral forms, 2 non-superimposable mirror images. And this crystallization not only purifies the riboform, but it amplifies any inherent chirality that is in the starting materials. So not only can we purify the riboform, we can amplify the handedness, which we need to essentially get to homochirality to build our ultimate go, our ribonucleotides. And now for the twist. This is what was a little bit different about this. If we now take our free riboaminoxazolene, sounds good, it's ribo, right? That sounds like we're moving towards ribonucleotides. What we find is that equilibrates with the arabinoaminoxazolene, so our other form that was part of our 70% mixture. And again, so this has only one stereochemistry. This has this position here is the wrong stereochemistry. This one has this position that's the wrong stereochemistry. If we treat our arabinoaminoxazolene now with another really simple organic molecule, 3 carbons, 1 nitrogen, we find that we build an anhydronucleotide, and this is a crystal structure of that compound. And if you're familiar with nucleotides, you'll see that it's very closely related to a ribonucleotide. We've built a cytidine ring. We've attached the correct nitrogen. We've got 1, 2, 3 of our stereochemistry right. We just need to invert this stereocenter. This is anhydro, so we've removed water relative to the nucleotides you find in biology. So that activates this position to inversion, and then something spectacular happens. If we take this compound, the product of these reactions, and dry that with the phosphate that's been essential to give the selectivity that we desire, and urea. Now, urea is the hydrolysis product of our first compound, so this is cyanamide again. Any excess cyanamide in our first reaction is slowly, and in a reaction catalyzed again by phosphate, converting into urea. If we dry this mixture down, what we find is this hydroxyl here is selectively phosphorylated. And that phosphorylation, that selectivity, is inherent to this compound. Usually a 5' hydroxyl, a primary hydroxyl, is less hindered, is freer to react than a secondary hydroxyl. But in this specific compound, what we find, and this is a crystal structure of that compound, and this is a molecular dynamics calculation, so a computer simulation of the bonds in that compound. What we find is there's a kissing interaction. The 5' hydroxyl that would ordinarily be more reactive than the 3' hydroxyl is touching the nuclear base. It's giving away some of its reactivity to the nuclear base, and this protects both the nuclear base from hydrolysis and the 5' hydroxyl from phosphorylation, and leaves this 3' out on a face on its own to be easily phosphorylated. We phosphorylate that position, and then this molecule rearranges. It undergoes an intramolecular rearrangement. The phosphate attacks this position here and inverts the stereochemistry. This does 2 things. It liberates the free cytosine, the free nuclear base that we need to transfer information in this molecule, and it generates the right stereochemistry, so up, down, down, up, the natural stereochemistry of ribonucleotides. Now, as a last and a bonus, really, and we weren't expecting to find this, what we found is if we took our 2', 3' cyclic phosphate that we'd made through this chemistry and then shined light on it, UV light, what we found is it was inordinately stable. You'll be familiar with DNA damage through the irradiation of UV light. This is how we get skin cancer. But these compounds, again, and I won't explain why because it's quite complex, but due to their specific nature, because they are specifically the natural nucleotides, have unprecedented photochemical stability. So over a really long irradiation, 82 hours, blasting these things with UV light, we find that we get about a 50-50 mixture of our starting material and a second ribonucleotide we need to build RNA. The benefit here really is not that we can make this compound, is that all the other isomers, and this is the most closely related, so this is just one stereochemical change from our product, are completely destroyed by this process. The stability that's gained by having this specific stereochemistry is only for this stereochemistry. So this gives us a mechanism not only of synthesizing these compounds, but purifying them from other compounds that might form Watson-Crick base pairs and get involved in information transfer. And what this allows us to draw out now is a 1, 2, 3, 4-step synthesis with purely chemical species of one ribonucleotide, its photochemical conversion to a second, and their purification by the use of light. And I will just remind you, because I think this is just an awesome thing to be thinking in 1863, how closely this relates to what Darwin was thinking. Ammonias, he means nitrogen compounds, phosphoric salts, heat. All these reactions are obviously done at room temperature or thereabouts. There's some heat present, and there's light present as well. I've dropped the electricity bit. I'm not sure that's important here. But we're essentially chemically forming a compound that's essential to biochemistry, to life and information transfer. And these processes allow us to tick off many of the requirements we had for synthesizing the correct compound. We partially get around this problem. We don't break chirality, but we can amplify EE on the way. This is a current target, I guess, for us, and something we can drive forward to understand the origins of homochirality, absolutely. But we get most of it. We don't quite get regiospecific phosphorylation. Now, phosphorylation is regiospecific, but we make a monomer, not the polymeric material. So I'd like to just touch on how we can use these monomers to address these two problems 7 and 8. And I don't have time for 9 and 10, but we can do some of this as well. So we can show that we can take chemical compounds and chemically assemble ribonucleotide monomers, the basic repeating unit of RNA. Can we chemically, without the requirement for polymerase enzymes, also get this backbone, this specific backbone that's found in RNA? So this is our monomer. It is activated with respect to RNA, a linear polymer, and it will slowly hydrolyze in water to give a mixture of 2 phosphates and 3 phosphates. We can quantitatively reactivate these, treating them with a compound like cyanoacetylene. We saw that earlier in our pyrimidine synthesis. But unfortunately, if we take these compounds and just try and polymerize them free, we get a mixture of these isomers, 2' and 3', so natural and non-natural. But more damningly, if we try and build a polymer using this activation with a template, so we try and transfer information from one piece of RNA to a second by building a new strand of RNA, just like biology would when it replicates DNA, we find damningly that we get absolutely the wrong stereochemistry. And lots of groups have explored this. It is absolutely wrong. Can we address this chemically, or do we need some interference from biology? Do we need proteins to assemble this compound in the correct form? And again, I wouldn't be telling you all this if we couldn't do it. What we can do is take our mixture of monomers, our 3' phosphate, essentially the one we want, and our 2' phosphate, the one we don't want, and treat these not only with our activating agent, which would have cyclized them back to the 2,3 cyclic phosphate, but also with a thioacid. So again, a really simple, prebiotically, abiotically plausible compound. And what we find is something special. This mixture differentiates our 2 monomers. The natural form is acylated. We add an acyl group to the hydroxyl. Nothing is added to the non-natural form. This isn't a magical process. It's very straightforward. What we have is a thioacid, which is much more nucleophilic. It's much more reactive than our phosphate monoesters. This reacts with our activating agent really, really quickly to give us an activated thioester. This activated thioester then acylates our phosphates. And then because these are different structures and they have different orientations, they sit in different forms, they undergo different reactions. The natural form transfers the acyl group to the 3' hydroxyl. The non-natural form largely undergoes hydrolysis, partially cyclizes back to the 2', 3' cyclic phosphate. Being able to achieve this reaction robustly in water and highly selectively allows us to come at this oligomerization problem from a different angle. We can now take this mixture of monomers, or indeed even polymers, where we're building them with template direction so that we can transfer information, and we can now build a polymer that contains the natural sugar phosphate backbone with an acyl group, which will slowly hydrolyze away to give us natural 3', 5' ligated RNA. Our residual 2-phosphates are recyclized because they weren't phosphorylated, and they form our 2,3 cyclic phosphates again, and we can put them back into the process to generate more 3', 5' RNA. And this allows us to solve another series of our problems. We still haven't quite solved homochirality absolutely, partially, but we can get to 3', 5' linked RNAs with good selectivity based upon chemical reactions and chemical control. And I think this goes a long way to realizing Darwin's dream of synthesizing a protein and a compound essential to biology through purely chemical means. But if we really want to synthesize life, we don't just want a single compound. Although RNA is important, it's not all there is to life. And this compound must be ready to go still more complex changes. So this is the challenge moving forward. This is what we seek to achieve. We can potentially make a protogenome, something that can contain information, something that can contain information in a way it's contained in biology. We can also show to some extent that these hereditable structures can then feed back into this process to influence its formation, and this gives us a feedback loop and therefore a selection pressure. If we have a genome that specifies a compound that, or a large polymer that increases the selection for synthesizing it, then we have natural selection. Natural selection can't really work without Nick's membranes. If we have this process in the absence of a membrane, without containing it, we just lose out to viruses. This compound, this genome, needs to keep its advantage to itself to realize evolution. If it synthesizes a compound that's going to increase its rate of synthesis, it doesn't want to give that advantage away to other compounds, otherwise evolution can't occur. So we need some form of compartmentalization to realize evolution, and we fully think we can build evolving organic compartments that can fulfill this role. And then finally, we need to get into our evolutionary arms race. We need to start thinking about how we can efficiently synthesize, better than any of our competitors, all of the components that we need to replicate ourselves. We need to think about how we can better take in nutrients and better pump out waste and more efficiently use the raw materials that are around us to build ourselves and build life. And this is then starting to think about how we can build a more and more complex metabolism based on the genome in our protocell so that this system can truly evolve and truly start to become a species like you and I, a real living cell. Okay, and that's where I will finish. I just want to thank everyone who works on this. We have a big team at UCL. This is not just my work. This is a lot of scientists who put a lot of effort into this. We have collaborators. Jack Shostak's already been mentioned. John Sutherland's another really important collaborator. And finally, our funders. We couldn't do any of this work without all these people who provide the money, the resources to let us try and realize solutions to this huge challenge. And then I'll just thank you guys for listening, and the organizers, again, for inviting me to this really fun night. And I guess we'll have questions. Yeah.